So welcome to Lunch with the Core. Next week's speaker is Dan Davidson from the CNRG. He's going to speak about changes to cloud data storage. This week, Dr. Kingsley Boateng is speaking to us about segmentation. So welcome, Kingsley.
All right. So thank you, and welcome to this presentation on how we can use some of the latest tools in segmentation to analyze microscopy data, looking at both optical microscopy and also electron microscopy. So oftentimes you will use an optical microscopy like LSM 800 or whatever to acquire some fluorescent images, but it is important that you are able to visualize this 3D data in an appropriate software. You may have to do some segmentation to be able to obtain some quantitative information about your data and maybe be able to interpret it in some context. Similar thing applies to grayscale data. It could be electron microscopy, MRI, or micro CT data. Here again, you obtain a series of stack of these slices. You may need to transport that into an appropriate software, do some rendering, and then further process this into some segmented data where you can actually obtain statistical information. So my goal is to quickly touch on some of the current tools that people are using, and then focus on two of them that I'm more comfortable with, and share some pipeline that I use in analyzing data. All right, so as a way of outline, I will be highlighting some current 3D tools. Basically, how we can apply machine learning in data analysis, focus on Imaris, and then Amira. These are the two main softwares that I will be highlighting today. And in particular, AI-assisted Amira deep learning and prediction. And then I will look at two applications of these and end up with talking about how you can create a recipe to apply this to a bunch of data, not just working on one data at a time. You can take different experiments at different times and just apply a recipe and you get your data out of it. All right, so there are tons of software that are currently available. A lot of them are free. In fact, about 80% of them are free. And they all have their strengths and weaknesses. Some of them are basically manual. You can just basically do everything manually. Some of them are semi-automatic. In other words, you need some human involvement to help you process the data. Others have been using deep learning, and now we have others that are focusing on using AI. And so there are a few common ones that people use, even including Fiji. And then you have some other tools that have been developed quite recently, like the Empanada. Keda was here a couple of months ago to talk about how you can use that to segment mitochondria from any source of mitochondria. It doesn't depend on organism, whether it's micro CT data or TEM data. You can use that to process mitochondria. There are others that have approached the same idea, but now they are developing a software. The Constantin Pape is aimed at being able to use the tool to segment a whole organism. So you are not even just interested in mitochondria. It could be the chloroplast. It could be whatever whatever is in the organism, you can segment it. So this is being developed by a group in Europe. Is still work in progress. And of course, there are several of them. And so most of these, you find them on GitHub. This is a database where developers put their software and people can test them and give their input. And so I've summarized a few of them and how they can easily be used. I don't have time to go through each one of them. But of course, there are also the commercial ones. You have the Arivis, Ameris, which I'll talk about. Zeiss came out with Interlysis. It didn't really catch a lot of attraction. But then we have the Amira and Avisio. These are much more common ones that people that are interested in commercial software use. So I will later on focus on one or two of these and go more into details. In fact, Facebook has also joined in the fun. And so Facebook has come out with Segment Anything. They call it SAM. And so originally it was designed to segment things in nature. You could segment buildings, you could segment anything. People have started applying it to biological samples. We tested it. You can actually go to the website, upload whatever file you have, and then it will segment it for you. This is only in 2D. And oftentimes the limitation is that you don't necessarily get any statistical information from this. It may do a good job of segmenting, but then you may have to then import it into some other third-party software to get information. There's a group that has tried to develop this further into what we call MicroSAM to aim at using this technology in analyzing cell data. So all these techniques, most of it is semi-automatic. There are a few of them that are now trying to use AI. So what is AI in terms of some of the context of what I'll be discussing? So we've heard about AI a lot. A lot of us know about it already. Basically, you are looking at algorithms that are able to sense, reason, and behave as if they are humans. And so that is the larger field of science. But then we have machine learning, which is a subset of that. And so here you have algorithms that are able to improve as you feed it with more data. And then you have deep learning. Deep learning is also another subset of machine learning, where now you are even going to train your model in a much bigger set of data. So it's not just one or two data. It could be tons of different type of data. And so models that are trained on such tons are basically, you doing this is what we call the deep learning. And basically the whole idea is that you are artificially training these algorithms to mimic the brain, the neural networks in the brain. And so that is the whole idea of deep learning. All right, so my goal for the next 20 minutes or so is to show you two of these software that I'm more comfortable with, and I think most of the time they do a very good job. The other advantage of these two software that I'm going to talk about is that they have everything in one package. For most of the free applications, you have to use it to do one aspect, import it to another software to do another aspect, import it back. It's not always one-stop shop with everything. And so the commercial packages are designed to have everything in one software. So that is why I'm going to focus on these two. But there are others that can do similar things, which are all free. All right, so let's start with Ameris. Ameris is originally designed for analyzing light microscopy data. And so you can import any kind of, it doesn't matter the type of microscope. It could be a Zeiss-based, Nikon-based. It can read into all the software from all the microscopes. Then you may have to prepare this. Maybe you have to do some filtering, and then you can render your data. And then, of course, you want to go ahead and analyze it. So this may involve doing some segmentation, doing some measurements, and then you may also need to interpret your data. Maybe you may do some post-processing. This is a very good tool for people that are doing tracking. It has some very nice tracking processing pathways that you can use. And of course, it's very easy when trying to do batch. So if your focus is only on light microscopy, I think Amaris is all you need. Amaris, of course, can also do grayscale data, which I will touch on in a few minutes, but its strengths in analyzing like microscopy data. The AI version is in the 10.1, so you have an earlier version, this will not work for you. The core has the latest version, which is the 10.1, that has the AI capability. So that is what I'm going to focus on. And so the AI version has been applied to cell biology, looking at segmenting nuclei and other things within a cell, looking at organoids, MRI data, and even EM data. So the strength of Amaris is that everything has been done to make it very user-friendly. Very user-friendly. And so you can go through just a few clicks, and then you have your data. And so the AI part is built into the Surface tool. So there are a couple of tools that you use in Amaris. You have the Spot tool, the Surface tool, the Cell tool, and then the Neural tool for people that are doing brain segmentation. But everything related to AI is built into the Surface tool. So you want to click on the Surface tool, and then you then go down here and select, you click in the next tab, and then you want to select the appropriate channel of interest, and then you want to select the machine learning segmentation. You click on the next tab, and then it brings you into this window. And all you need to do is that you define what is the background, what is the foreground. So your background, all you need to do is just draw some brush strokes of what you think is a background, and then you do the same thing for the foreground, just a few brush strokes. You have this tool here that allows you to move into other layers, and you repeat it. So if you have a slice of 100 slices, maybe every 20 slices you repeat this there. And then all you then do now is to tell it you try to train it on that batch of data that you've segmented. So like I said, you go through a few slices, repeat this, and then you just hit train. It is very, very fast. Sometimes it takes just about 10-15 minutes, and then it is done, because everything is done on slice by slice. And so it's a very fast tool when you're trying to segment this. And so obviously for the first attempt, it may not look perfect. So here we are trying to segment this nuclei, but it doesn't segment it completely. So you repeat, once again, you go through a few more layers, draw some brush strokes, maybe give it much more defined, and then repeat it, and then that's it. And so now you have your segmented data. Of course, you may have to do a few post-processing steps. Maybe you want to filter out smaller objects. So you can filter based on several criteria. It could be volume, it could be area, it could be intensity. And so you can play around with this histogram and remove objects that you think are not appropriate, and then you're done. And so this can be very, very fast. I tested this, I think within 10 minutes, and actually less than that, and I had a full segmented data. Something that in other software it would take me about 30 minutes, I could do this less than 10 minutes in Amaris. So that is the strength of Amaris. It's very, very fast, user-friendly. It is not complicated at all. So that is where you can start from, especially if you're dealing with light microscopy data. So this is our segmented nuclei. So the data that I just showed you is Drosophila eximba, and so what we just segmented is the big nuclei instead of the small nuclei. Now what you want to do then is you can do some other post-processing. Maybe you want to look at statistical information from each of these segmented objects. You may want to create a batch processing, a model for batch processing. So that is right here in the creation tool. So you can click on that, and then you store the model that you use in the batch. You can give it any name, but you can store it in the Arena or the favorite creation parameters tool. You click OK, and then it appears as this icon. So in the arena view, it appears as this icon. And then whatever other data that you want to apply this to, all you need to do is right-click and hit Run. And then maybe depending on the number of data that you have there, it may take 2 hours, it may take overnight or whatever, but then you don't touch it, and then it will output all the data for you. The other advantage of this is that for each data, it will put out an Excel spreadsheet that has all possible measurements as well as the actual segmentation. So you can go back to each data and look at the segmentation and then the Excel file and see if it makes sense. All right, so Amaris is very friendly and very easy to use. Amira does a very good job, but it's not always very user-friendly. And so a lot of people struggle with it, but I think it is much more powerful, and it has more, you have more control in Amira. Amaris, everything is done in the background, so you don't have too much control. But Amira, you have more room to try more variety. What I'm going to do is quickly look at the traditional deep learning in Amira, and then the latest AI-assisted deep learning. So all you need to do in Amira, first, obviously, you want to import your data. And so here I have these slices that I imported. And then you want to extract a subvolume to then train a model on. So you can use the extract subvolume tool. You hit apply. And so now I've extracted this subvolume out of the entire data set. And then what you then do next is that you go to the segmentation workroom. And in the segmentation workroom, you use the brush tool if you want to do manual segmentation, and you paint on the objects that are of interest. So here I painted a few slices of the mitochondria, and then you add it to the appropriate material. So first material that I created there is the mitochondria. And then you do the same thing now on the membranes, and you add it to, you create new material, and then you add it to the membrane. So I have two material created. And then what I want to do next is that I'm going to come back to the project view. And so this is the extracted prediction or the mask that I've created for the membrane and the mitochondria. And so what I'm going to do is then use this. This is from the subvolume. Use this to try and predict the rest of the data, or even develop this into a recipe where I can use this on multiple other data. So that will require a few steps. First, you want to build a model. And so to build a model, you start with the arithmetic tool. The arithmetic tool allows you to combine these two masks into one file. And the expression you want is A plus B times 2. What that means is that you have A as class 1, B as class 2, and these are separate. It is not merged as one single class. All right, so that is what that means. And so you can then, sorry, I've jumped a couple of steps. You can then go ahead and hit the next or apply. And so this is the result of the arithmetic tool. Now I load the actual data set that I want to try this on. The sub-volume that I want to link to the target. So I have a sub-volume. I have the mask for both the mitochondria and the membrane, and I want to link these two together so that I can use this to first train on the small volume. And so you right click on that, and then you choose the deep learning training segmentation in 2D. This is the one that you want, and you hit next. And then, so you have the deep learning tool now uploaded, and you need to define a few parameters. So first of all, you want to link it to the target, which is the mask. And then another important thing is you want to indicate the number of classes. So we have 3 classes: the background, the mitochondria, and then the membrane. So we put in a class of 3. The output directory, you define where you want the model to be stored. And then other parameters that you can play with is the patch size. This increases in 32. So you can have 32, 64, 96, 128, 256 in that order. You can also play around with the patch per image. Typically I use 4. You can try different numbers to see how it works. And then you hit next. And so it's going to show you the progress of the training. This will be a pop-up. And so this is the original data and this is the training. And so in the end, you see the accuracy of the training. And so you have the first class, second class, and then all the 3 classes. You also have a plot of the loss. So you want the accuracy to increase and then the loss to decrease. And so if you see this kind of trend, it means things are working fine. And so we have the model that is trained and is stored. Now, what we want to do now is to apply this model to the rest of the original data. We trained this on the subset. Now we want to apply this to the rest of the original data and see how we do. So what you need to do now is to load the deep learning prediction tool. So you right click on the image, the rest of the full set of data again. You right click on that and then you select your deep learning prediction tool. And so that is the deep learning prediction tool. And now you want to look for the model that you trained. All right, so in the architecture window, you click on this and then you go to where you save the model and then you open that and you hit next. And so within a few minutes, you have your prediction. And so this, depending on the volume of the data, it could be maybe 10 minutes, it could be 20 minutes, but oftentimes this is pretty fast. And so we have the prediction of the entire data set. And so just to give you a perspective, you can attach a color wash tool so that you can see the original and the segmentation. And so you see that it did a fairly good job. So we once again, just to recap, I have a structured subvolume. I created a mask for the structures of interest, and I use that to generate a model, and I use the model to then predict from the rest of the data. So here you can then process it further to obtain statistical information about whatever object that you segmented. If you are interested in doing more learning more about deep learning, there is a guide, a very good guide. And so you go to the help tool and then you can read more into this, but this is pretty easy to do. But what I've done here, you saw the steps doing a lot of painting and going through each slices. You can do that step with the AI-assisted deep learning. And so this can be done only in the latest version of Amira, the 2022.2 version. That is the latest version. And so once again, you import your data, and now you go to the segmentation workroom. Now all you need to do now is add patches. All right, so now you're going to add patches. So you hit on the create new patch set. You can define the size of the patches. So once again, it could be 96, it could be 128, it could be 256. So you define the size of patches, you add a couple of patches, and then within you want to add the patches to areas where you want that to be marked as a background, as well as the structures of interest. And then for the patches that are on the structures of interest, you need to make sure that you can use the paintbrush to paint those structures that are within the patch. And so the accuracy of the painting will determine how well the prediction is going to be. The brush can be sometimes very slow, so there are other semi-automatic tools like the Magic Wand that you can use, and you can do this on a particular plane. You move through the slices, you do this. So if you have a stack of 100, maybe you do every 20, you do about 5 layers of that. And then now you come to the machine AI-assisted segmentation workroom. So you click on this tab. You want to define your iteration. So iteration here is defined as epochs. And so it could be 20, it could be 25. And the higher the number, the longer it will take, but the more accurate is your prediction. You also want to define your elastic deformation strength. So for straight objects, you want smaller numbers. For things that are more curved, you can increase that number. And then you hit create selection. And once again, it's going to then go through and segment the entire thing for you. Now, once that is segmented, the segmented object here is not transferable. That is the challenge here. So in the AI-assisted tool, once you use the patch to segment, you cannot use that directly on another set of data. So for us to be able to use that training to apply to another set of data, we need to extract the patch. So the patch will be similar to the extracted subvolume that I used in the deep learning. So we need to, once again, go to the data and extract a patch out of the entire data set, and then retrain this patch to be able to predict. So that is what we're going to do. So you hit on the extract patch. You want to link that to the particular patch set that you use. So in this case, I initially tried 2 different patch and I like the second one. So I linked that to the second patch, and then you hit next. And so this is the extracted patch that I'm now going to use to generate a model that I can use in predicting or segmenting other data sets. So there are still a few steps that you need to follow after you've been able to extract your patch. Now you have to once again go in and then right click on this and select the deep learning segmentation in 2D. This is the same thing that we did with the deep learning approach. And so we're going to repeat the same steps even with the AI assisted approach. The AI part only helps you to use the patch in doing your selection. The rest of it is the same process. And so now I've created my deep learning training segmentation 2D tool. Now I need to go in and define a few parameters. Once again, I want to link it to the extraction, and then define the parameters like the number of classes. Here I have only 2 classes. One, I have the membrane or the myelinated axons, and the myelination, and then I have my background. So only 2 classes. And then I need to also define my output directory where I want my model to be saved. You want to, once again, refer to the particular size of patch you use. So in this case, I use 256. So you put in the same number. The patches per image is 1. And then images per batch. This can be variable. You can play around with this and see, but typically 4 is fine. You hit next. And so once again, you see a prediction the progress of the training. And so once that is done, the training model is stored. And so you have to then again test this on your data set. So now again, you go back to the deep learning prediction model. And so you go to wherever you saved your prediction. So in the architecture, you select that. And then you open and then you hit next. And so this is my prediction using the AI-assisted model to predict for the rest of the data. So this part is quite straightforward, but oftentimes that is not the end. And so depending on the type of application, you may segment, but you may have to do some post-processing. So oftentimes you can, at this stage, create a recipe that you can use for batch processing, and I will leave the recipe part to towards the last end of my talk. But I want to go ahead and show how you can use the AI-assisted tool in segmenting two applications. I want to talk about the C4 plants that Don Oz Group works on, and the myelinated azones that Stillman's Group work on. All right, so the C4 species are very important for human food and also as a good source of energy. So we are looking at maize, sorghum, and sugarcane and crops like that. And so the size of the chloroplasts and the other substructures are important for the efficiency of photosynthesis. And so what labs that are studying this have been trying to do is to try to do measurements. And typically in the past, these measurements have been done only in 2D. Obviously in 2D, you can have biased information or a little bit less quality data. And so the goal is to then apply this in 3D volume sets to measure chloroplast volume and other structures like that, plasmodesmata. And so last year, we published a paper that has done this work using a mirror, but almost everything was done using the semi-automatic approach. And so if you talk to Lee Munsab, which is currently a professor back in Hong Kong, he will tell you how much time it took for him to work on this data. But now we have a way around this that it will take much, much less time. All right, so all that you need to do first, I forgot to mention that for the AI to work, you need to first create a Python environment. So you go to Python and you can tell it to create a Python environment. It takes about 30 minutes to load a Python environment. Then you are ready to use the AI version. And so once again, you load your data set. You can extract a cell volume. And then as I showed you a minute ago, you create patches. All right, you create patches. Either use the hand brush or these other semi-automatic tools to paint the structures of interest. And then you go to the AI-assisted workroom. You are able to then tell it the number of iterations, the deformation, and then you create selection, and it's able to predict for you. This can be done within like 10 minutes, and then you're done. It's very, very, very fast. And so Lee, before he left, he tested this. And so this is AI-assisted segmented data for both mesophyll and bundle sheath cells. And so compared to the one that he did, the publication, this you can do under 30 minutes. This took 5 hours to days. And so the advantage of this is that not only are you doing this in shorter time, you can then also do it in batch. So you can apply it to a batch and just give a command, go walk away, and maybe overnight it's going to do hundreds of these for you. All right, so you don't have to sit by the computer. So that is the advantage of the AI-assisted tool. Moving along, I'm going to briefly touch on another application. So the myelinated ozone G ratio is important for people that are studying the transmission of information in the brain. And so G ratio is basically being utilized as a functional and structural index for optimal axon myelation. And so the thickness of the axon and the size of the myelination are all very important, and the variation will affect how information is transmitted. Maybe sometimes even diet or different factors can affect the size. And so labs are interested in this. So it has been predicted that for very efficient transmission of information, the G ratio optimal should be about 0.6. But depending on the organism and even the type of area of the brain, the G ratio may vary. And so for the mouse corpus callosum, you have a range of 0.76 to about 0.8. Now, traditionally, this work was done doing microtome you do slide sessions and you do tolerance staining, and then you sit down and manually do these measurements. But with the advent of high resolution techniques like the serial bulk phase SEM, you can get much higher resolution images. And so here you have a wild type and knockout. You can clearly see that there's a differences in the size of the myelination. And so then you can do these measurements. So the G ratio is basically a ratio of the inner diameter over the outer diameter. And so depending on your the myelinated axon, maybe it is a sphere, maybe it's elongated, you can apply these measurements and calculate your G ratio. And so about 2 years ago, I was involved with working with Allison. We spent a lot of time trying to explore tools to use this. And I thought Amira was perfect, but in the end, the lab resorted to much more manual methods in doing that. I'm yet to understand the reasoning. But we spent a lot of time working on this, and we could actually obtain statistical information. You could calculate G ratios, which we actually could validate. And so this is image A, process data. This is 2D. You draw a circle in the inner, outer, and you do your measurements. And then we compare that to the Amira segmented, semi-automated segmented, and we see that the data is very comparable. But like I said, that has not been adapted in the lab yet, and hopefully, based on what I'm going to talk about today, labs and similar labs can consider adapting this new approach. Currently, what most people do is they use mile tracer, and this is a purely manual segmentation. You draw a circle, or you paint the inner and then the outer, and tell the software which part is the myelin and which part is the axon, and then it will do the calculation for you. But this is, like I said, is very laborious. You have to do this one axon at a time, and it can take hours to do even just a few of them. So I'm going to try and quickly go through a pipeline or workflow where you can do this in a few minutes, and this can also be done in batch. All right, so once again, you load your data, you go to the segmentation workroom, and then you apply the same patches that I talked about earlier on. So I did that to segment the outer membrane, which is the myelination. And then after that, what I did was then I went to the mask that I've created for the myelin, and then selected that, and then I filled the hole. All right, so this is the fill hole creation tool. You fill the hole, and then I locked the myelin and then added the hole into a new material. So I have a material that is just the myelin as well as just the axons. And so now I go through and do a few steps to process the data in a way that can be used for batch processing. Well, the first thing that I want to do now is to apply the threshold by criterion tool. What this tool does is that it splits the data into two classes, because I want to obtain measurements on the two. I want to obtain measurements on the inner, which is essentially the axon, as well as the outer myelin, and so what I need to do is to be able to separate these two. And so the threshold by criterion, you can tell it to equate or subtract. So this is the class 1 material, which in this case is my myelin. And so I have that data set, and then I can obtain measurements on this. And then I do the same thing for the axons. And so now I want to define the kind of parameters, the measurements that I want to do. So you go to the measurement tab here, and then the tool that we are looking at is the label analysis tool. So you import the label analysis tool. You click on here and you define the parameters. So there are some predefined parameters that you can use, but oftentimes it may not necessarily have what you need. And so you can create your own and go through a series of different measurements. And so here I added a couple of measurements. And the way you do that is that you can go to the Hub Guide, type in length or measurement, and it will give you a list of measurements, and you can read about it and see if those are meaningful. So 2 of them that I was interested in is the length is a type of ferrite diameter where it measures the longest diameter of whatever object you're interested in, as well as the shortest diameter. In actual fact, is the length that I'm interested in. So I incorporate that into my measurement tool and I apply that. And so now I have measurements for the axon only. You can have several other measurements in there. I now need to do the same thing for the myelin, but that is not so straightforward. So you see that a lot of them are touching, all right? And so this is all one object. I need to find a way to separate this. And so to separate this, I first need to fill this so that now I have a full object, not just a circle, right? And so once again, I combine the 2 together using the arithmetic tool. And now I have these 2 combined, and now I need to make this as a single mask, not a 2-layer mask. And so I went through 2 more steps. I threshold that threshold by criterion, set it to 0, and so that is what I have. And so now I have just the combination and then the background. So I inverted it so that I have just the combined myelin and axon. And now I can now split the touching objects. And so in Amaris, in Amira, there is a split object tool that you can use to split objects. It does a fairly good job, but sometimes it struggles. And so this is supposed to be 1, but it's split it. Sometimes, like these areas, it may struggle, but to some extent, it works at least 80 to 90% of the time. It does a very good job. Right after this, if there are things that you think are not of interest, you can filter them out like these. So I applied a filter by measure tool to filter out things that I think are not important. And so I compared the splitting tool in Amira to the splitting tool in ImageJ. So I imported the same data to ImageJ, split it using the water shell splitting tool, and it does just about the same, maybe slightly better, but I wanted everything done in one software because I want to generate a pipeline for batch processing. So maybe this is 5% better, and so that I went back to Amira and then followed along with the separated data and then did my measurements. So now I have my measurements of this then gives me the outer diameter measurement, all right? And so now you can export this into Excel sheet. Within Amira, you can sort these out. You can use the sort tool, align them, run them in maybe from the biggest object to the least object. You can use the search tool to look at a particular one and make sure that the numbers make sense, right? So you can validate your data. And so I have the measurement for the inner, which is the axon, as well as the outer one. And then I export this into Excel, and then I do my G ratio measurements. And so I can then validate the data and make sure that the numbers that I'm getting make sense. So once I'm done with that, now I want to create a recipe for batch. And so within the next 3 minutes, I will touch on that and I will conclude. I know I'm going a little bit behind time. So once again, to create a recipe, you go to your final result. So this could be your label analysis, or this could be your segmented data. All right, so you go to your final result, you right click on that, and you create a recipe. And this is the recipe creation workroom. You can edit out steps that you think are not so relevant. Maybe you did some steps that you think are not important. You can edit that out, and then you save it. Once you save this, now you have to be able to use the recipe player to play this recipe on a different set of data. Now, the recipe player doesn't come with the Amira, so you have to install it. So you need to go to the extras room and then install the recipe player. And so after you do that, you right-click in the window and then load the recipe player that we just downloaded. So that is the recipe player that we just downloaded. Now, I'm now going to load the recipe that I've created and try to process it in a bunch of files. So once again, once you click on the recipe player, you can define a few parameters. First, I want to load my recipe file that I created. And then I also want to load the files that I want to process. So you could have hundreds of different files that you want to process. You just load them at once. And then you open, you click OK, and then you add. And then the last thing, you want to define the output directory where you want it to be saved. So oftentimes, it might be good to create a folder in advance. But if not, it can only allow you to have a new folder. So remember, the new folder is your output directory. So once again, you choose the file, and then it will show you the progress. So maybe if you have hundreds of files, you leave it overnight. By the next day, you come and it's done. And then you can visualize your data. So the data is in the form of an Excel file, which can be opened in Amira or in Excel. In Amira, you can load the Amira version and click show, and then it will show you the data once again. And so you can obviously also open the Excel file in the spreadsheet and then do your validation. And so this is, it looks like a few steps, but it can save you time because you can then apply this to batch. And so to conclude, there are further resources that you can fall on if you're interested in using Amaris or Amira that you can fall on. In Amaris, all you need to do is just a few brush strokes, and you can segment. I tested this same data set even in Amaris, and it does a fairly good job. Maybe not as good as Amira, but you can actually use it on grayscale data. Obviously, Amira is a much more reliable tool when it comes to grayscale data. And so whichever your choice, you can look into that. And with that, I want to thank you for the attention.
Questions?
So in terms of accuracy, so if you do a head-to-head comparison with somebody doing a manual or semi-manual segmentation who's knowledgeable about the data set, how does Amira compare? I'm thinking about the EM specifically.
Yes, it depends on sometimes how much your model is well trained. But if you spend time ahead in training a model very well, it's sometimes about 80 to 90% comparable to the manual one. Yes.
This was great talk. Thank you very much. Is the model that you create can be used for future data?
Yes, future data. All you need to load the recipe and hit play, and then it will do it for you. Yes, yes, yes. Before you load the data, you have to do pre-alignment. Yes.
Is Elastic a tool, or do you know any other tools that does that?
Amira has an alignment tool. Oftentimes, I do that in the imaging software that I use in doing the DMS3 software. I do that, but Amira also has a tool to do the alignment. Yes.
So for that one example of the axons, there was like one image you showed where the axons were different colors, sort of like an instance segmentation example.
Mm-hmm.
I was wondering how you would do that through Amira as opposed to just saying, like when you're making your masks, instead of, like, would you have to color each cell a different color then?
No, it's going to be the same color. I only just use that to highlight it, but it's going to come as the same color.
Okay, what
Yes.
if you wanted to do analysis like comparing each individual cell to each other, even if they're like all of one cell type?
So after you finish the labeling tool, for each specific item, it will give you a color for it.
Oh, it will.
So there's a color code for each item. So you can go to that particular one and compare that to the actual statistical measurement that you did.
Okay.
Yes.
I just have a quick question, because it happened to me in the past that after I obtained, light sheet microscopy images, and then I transfer those data into Imaris, I have a lot of background, especially in the green channel. So the question that I have is, like, what is the best software in order to get rid of the background? And if there's any specific setting that I can use in order to extract the background and proceed with the quantification of the image.
Yeah, so that is a special case for light sheet. Oftentimes, because of that problem, we recommend using other wavelengths. So basically, you want to do maybe 647 dye.
Yeah, 488, for instance.
Yeah, but 488 usually struggles because in light sheet, basically, you get a lot of more background in 488 than any other ones. So that is an endogenous problem in the light sheet setup. And so you can play around with it, but ideally, to avoid that completely, if you can, use higher wavelength labels. Yes, that is the easiest fix, yes. So the patches is just a sub-volume to train that model on it. And then you can use that patch to then train a bigger set of data, and then you create a deep learning model from that, which you can then use it for batch processing.
So this does need a training, but do Imaris, so Imaris doesn't.
Amaris doesn't use the patch. Amaris just use brush strokes. That is all. In the Amira, instead of the brush strokes, you still paint, but you paint within the patch. Yes.
So let's thank Kingsley again. If you have extra questions, feel free to talk to Kingsley.